Hello friends, in this video I'm going to start with this very important topic, glycolysis. I'll be taking glycolysis pathway in a story form so that you are not cramming all the steps. In this story, you will be knowing that why the next step is occurring. So that's why it will be very easy to understand. And do watch the basics of carbohydrate metabolism video before taking this glycolysis video, right? So let's get started. Glycolysis, also known as Embden-Meyerhof pathway by the name of scientists who have given it this, also known as EMP pathway, short form EMP pathway. So you know HMP is hexose monophosphate pathway which produces NADPH, that is a minor source, that is a minor pathway of oxidation of glucose. But glycolysis, EMP pathway is the major pathway of glucose oxidation. So even if EMP written in the option, so you should be knowing that this is glycolysis written, right? So let's start. The first step is glucose gets converted to glucose 6-phosphate. I told you the first step which occurs is always phosphorylation, and this is because of this is for the entrapment of glucose inside the cells, right? So this first step is phosphorylation, glucose to glucose 6-phosphate. Here, for adding this high energy phosphate bond, ATP is used. ATP is used, gets converted to ADP. The enzyme must be some kinase. I told you whenever something with ADP, ATP, mostly enzyme is a kinase, right? So here ATP used, enzyme is kinase. Here it is hexokinase or glucokinase. The difference between hexokinase and glucokinase we'll be later doing in enzymes topic, right? So the first step is done, hexokinase, ATP is used. Why is glycolysis occurring in cells? The reason is to obtain ATP. But at the very first step, we are using ATP. We are doing opposite of the target. Like when opposite of the target is done, this is known as rule of business. Because, see, in business, we want money, right? But at the very first step, we have to invest money. So only first when we invest, later we'll be able to gain. So glycolysis is also like a business that first we are adding ATP and after that we'll be obtaining ATP. And any good business is that where we invest less and we obtain more, right? So I'll be taking glycolysis as a rule, as a plan of business. The nature has made this plan of business. So let's see how. The second step is glucose to fructose, glucose 6-phosphate to fructose 6-phosphate. Because glucose and fructose are isomers of each other, so here the name of enzyme is isomerase, simply, right? And in the next step, fructose 6-phosphate gets converted to fructose 1,6-bisphosphate. So here we are adding one more phosphate at position number 1 now, making fructose 1,6-bisphosphate. Phosphate is a high energy bond. To add phosphate, we always use ATP. So here also ATP used, gets converted to ADP. Enzyme must be some kinase, and that is phosphofructokinase 1. Short form, this third number step, short form PFK1, phosphofructokinase 1. This is the rate limiting enzyme of glycolysis, also the first committed step of glycolysis. What do you mean by committed step? Committed means when this step will occur, then it is very sure that glycolysis will occur. That means first and second steps of this pathway were not committed for glycolysis because glucose 6-phosphate and fructose 6-phosphate, they can be used for other pathways also. But fructose 1,6-bisphosphate, when this compound is formed, fructose 1,6-bisphosphate, then we are very sure that now glycolysis will occur, pyruvate will be formed, right? So therefore I say PFK1 is the committed step, first committed step of glycolysis, right? Now let's see the fourth number step. Fourth number step is done by enzyme aldolase A. Write down A here, aldolase A. Where is aldolase B? Aldolase B will be doing later in fructose metabolism, right? Aldolase A is in glycolysis. The substrate for aldolase A is fructose 1,6-bisphosphate, whereas the substrate for aldolase B is fructose 1-phosphate, right? Let's see what aldolase A is doing. Aldolase A will break down this compound. This is a 6-carbon compound with 2 phosphates, which is now broken down to 2 3-carbon compounds. One is aldehyde sugar, other is keto sugar. Both are given 1,1-phosphate each. So let's see. Fructose 1,6-bisphosphate gets converted to dihydroxyacetone phosphate. This is 3-carbon keto with a phosphate. dihydroxyacetone phosphate. the short form DHAP. Later I'll be using the short forms. DHAP is formed. On the other hand, 3-carbon-aldehyde is glyceraldehyde 3-phosphate. So this 3-carbon-aldehyde is also given 1-phosphate. So now we have got DHAP and glyceraldehyde 3-phosphate as the end products of number 4 step. Now number 5th step is DHAP also gets converted to glyceraldehyde 3-phosphate. This way we get 2 molecules of glyceraldehyde 3-phosphate, and the name of 5th number enzyme is this aldehyde and keto interconversion is always by isomerase. So number 5th enzyme is isomerase. Now see this glycolysis pathway as a whole. There are 10 steps in glycolysis. These first 5 steps we are done. These first 5 steps constitute phase 1 of glycolysis, also known as energy utilizing phase of glycolysis, or you can say investment of business. This is the investment of business, right? And phase 2, the next 10 steps come in phase 2, and that is energy producing phase of glycolysis. Energy producing phase, right? Now see one smartness planned by nature. I told you that any good business is that we invest less and we obtain more, right? Now see phase 1. In phase 1, how many molecules are entering? How many glucose are entering phase 1? Only 1 molecule of glucose is entering. And therefore, whenever we use ATP in phase 1, we have to use it for 1 molecule. But in phase 2, how many molecules are entering phase 2? 2 molecules of glyceraldehyde 3-phosphate are entering. So that in phase 2, when we obtain energy, then every energetics is multiplied by 2 because 2 molecules are entering. This is a smartness of business planned here, that investment is less but we obtain more energy. Now let's see the detail of phase 2. The first step in phase 2 is number 6 step, that is glyceraldehyde 3-phosphate gets converted to 1,3-bisphosphoglycerate. This is the master plan of glycolysis. This step is very special step in glycolysis. Understand this very carefully. See here, glyceraldehyde 3-phosphate is getting converted to 1,3-bisphosphoglycerate. Can you see in this step one more phosphate is coming at position number 1? This phosphate, we are adding phosphate, high energy bond. Usually to add phosphate we use ATP, but in this step we are not using ATP because we are in energy producing phase, we are in phase 2. So we will not use ATP here. So this phosphate is simply taken from inorganic phosphate. Inorganic phosphate is always there in the medium, so we can take it. But generally, I repeat, generally whenever we add phosphate we use ATP. But this step is very special step where ATP is not used but phosphate is added. This is a very special and rare step which can add phosphate without using ATP, right? And the name of this enzyme is glyceraldehyde 3-phosphate dehydrogenase, and this is the only step of glycolysis which can use inorganic phosphate. And see, the name of enzyme is dehydrogenase. I told you whenever dehydrogenase means hydrogen removed and somebody will come to take this hydrogen, that somebody most of the time is NAD. Here also it is NAD. NAD will take this hydrogen, forms NADH. I have written 2 here because we started with 2 molecules of glyceraldehyde 3-phosphate. These 2 NADH will later go into ETC to give 5 ATPs. So this step is responsible for giving 5 ATPs. See, this is such a special step. In this step we are adding phosphate, and to add phosphate we generally use ATP. But this step is not using ATP. In fact, it is producing ATP, and that too 5 ATPs. Till now, we have just used 2 ATPs and we have obtained 5. It is such a smart plan of business, and many more ATPs are yet to come. Now let's go further. We have reached till this point 1,3-bisphosphoglycerate. Now this 1,3-bisphosphoglycerate is a 3-carbon molecule having 2 phosphates. Now only one work is left in glycolysis. That is, we have 2 phosphates. Phosphate is a high energy bond. Now we will keep breaking these 2 phosphates one by one, and when we break high energy bond, then energy is released. That energy can be trapped in the form of ATP, right? Only this work is left. So let's see. 1,3-bisphosphoglycerate gets converted to 3-phosphoglycerate. So you can see we are removing phosphate from number 1 position. 1,3 gets converted to 3. We We are removing phosphate from number 1 position. Who will take this phosphate? ADP will take this phosphate, gets converted to ATP. I have written 2 here because we started with 2 molecules. And whenever ATP formed at some step, immediately there and then only ATP formation occurs, known as substrate level phosphorylation. And enzyme of substrate level phosphorylation is always a kinase. And here it is phosphoglycerate kinase. Short form is PG kinase. PG kinase is a substrate level phosphorylation step of glycolysis. They ask you many questions on substrate level phosphorylation steps. So it is very easy to learn these steps. You have to learn the substrate, you have to learn the product, you have to learn the enzyme of such a step, right? Now we have reached at this point 3-phosphoglycerate. Now we have our last phosphate attached at number 3 position. We can remove this phosphate also and we can obtain energy. But there is a problem. When 3 number phosphate is attached at glycerate, this is not a high energy bond. This is not a very high energy bond. If I break this bond here, then I won't be able to get enough energy to make ATP, right? So what we have to do is that first we have to make this bond high energy and then we will break this last high energy bond. So our next 2 steps, that is number 8th and number 9th step, is like a preparation to make high energy bond. So let's see the detail. 3-phosphoglycerate gets converted to 2-phosphoglycerate. So phosphate is getting transferred from number 3 position to number 2 position. And the name of enzyme is mutase. So what is the EC number of mutase? Transfer of phosphate is occurring from position number 3 to 2. But mutase is not EC number 2 transferase. No, it is not transferase. Because in this step, molecular formula is not changed. One isomer is getting converted to another isomer. Transfer is occurring. But within the molecule, intramolecular transfer is occurring. And whenever in cells intramolecular transfer is occurring, be it methyl group, sulfate group, phosphate group, the enzyme used is always mutase. And mutase belongs to isomerase category, that is EC number 5, not EC number 2. Be careful with this. Now next step, 2-phosphoglycerate gets converted to phosphoenol pyruvate. Enol. Focus here. En means double bond, ol means OH. So this kind of bond with double bond and OH is made here. So you can just take an idea that this is a high energy bond. And to make this enol high energy bond, which enzyme is used? Enolase, right? So to make enol bond, it is enolase enzyme, very easy, right? Now, phosphoenol pyruvate we have obtained, that is we have our last phosphate attached to pyruvate with high energy enol bond. We can break this enol bond and we can get pyruvate. On the other hand, phosphate is taken by ADP, gets converted to ATP. I have written 2 here because we started with 2 molecules. Whenever ATP formed at such a step known as substrate level phosphorylation step, the enzyme must be a kinase, and here it is pyruvate kinase. Here it is pyruvate kinase. Now pyruvate is the end product of aerobic glycolysis, but lactate is formed in case of anaerobic glycolysis, and here the enzyme is lactate dehydrogenase, short form LDH I have written, lactate dehydrogenase. Can you tell me where is pyruvate dehydrogenase? Pyruvate dehydrogenase is in link reaction, right? But this lactate dehydrogenase is in anaerobic glycolysis. So we are done with the pathway and all the 10 to 11 enzymes we are done. Now I'm going to write the EC numbers of all the enzymes. See, hexokinase EC number is 2 because it is a kinase. Isomerase EC number is 5. Then PFK1, it is a kinase, so EC number 2. Aldolase A, it is breaking the bond without using water. That's why EC number is 4, right? 4 is lyase, bond is made or broken, water and ATP not used. So aldolase A or aldolase B both are in EC number 4 category. Then 5th number enzyme is isomerase, so EC number is also 5. Then 6th number enzyme is dehydrogenase. Dehydrogenase means EC number 1. Then PG kinase, so 2. Mutase, 5 I told you. Enolase is EC number 4. This I will explain later in enzymes topic. Then pyruvate kinase, because it is a kinase, so EC number 2. And the last one, lactate dehydrogenase of anaerobic glycolysis, EC number is 1 here, right? Now there are 3 irreversible steps in glycolysis. These are, you can see here, broad black arrows I have made. These broad black irreversible arrows I have made. So these three enzymes, these three steps are irreversible or regulatory steps of glycolysis, that is hexokinase, PFK1 and pyruvate kinase. So you can note here that all these three are kinases, right? Hexokinase, pyruvate kinase, phosphofructokinase. So these three regulatory steps are kinases. Now there are total 4 kinases in glycolysis. One extra is PG kinase. So can I say that all kinases of glycolysis except phosphoglycerate kinase are irreversible or regulatory steps of glycolysis? And now let's see the substrate level phosphorylation enzymes of glycolysis. That is this and this, that is PG kinase and pyruvate kinase. They are substrate level steps of glycolysis. Now you can see pyruvate kinase enzyme is coming in the list of irreversible regulatory enzymes also, and pyruvate kinase is also substrate level phosphorylation enzyme. This enzyme is common in both the list. I told you something special about pyruvate kinase enzyme. You have done that. Pyruvate kinase enzyme requires potassium more than magnesium. Most of the kinases, they use magnesium, but pyruvate kinase requires potassium more than magnesium. Now one more point about pyruvate kinase is that this enzyme deficiency is the second most common human enzyme deficiency. There is one more enzyme which is the first most common human enzyme deficiency, that is G6PD enzyme, which is enzyme of HMP pathway. Pyruvate kinase enzyme is of EMP pathway or glycolysis. G6PD is enzyme of HMP pathway, and this is the first most common human enzyme deficiency. Now both these patients, that is pyruvate kinase deficient and G6PD deficient patients, they both are very common. See, first most common and second most common enzyme deficiency. They are common and they have one common clinical feature, that they both present with hemolysis. So how will I distinguish between these two patients? There's a simple thing, Heinz bodies. You know, Heinz bodies are present in G6PD deficiency. So if hemolysis and Heinz bodies are present, then it is a G6PD deficient patient. If hemolysis is present but absent Heinz bodies, then most probably it is pyruvate kinase deficiency. Now let's see how hemolysis occurs in deficiency of pyruvate kinase also, and why hemolysis occurs in G6PD deficiency. In G6PD deficiency, hemolysis occurs due to oxidative stress, because normal role of G6PD in RBCs is to make NADPH, which will make reduced glutathione, which will destroy H2O2. But if G6PD is not there, then H2O2 is not destroyed and it will start accumulating in RBCs, leading to damage to the membrane of RBCs, leading to hemolysis, right? And why pyruvate kinase deficiency leads to hemolysis? Because pyruvate kinase or any enzyme of glycolysis, if deficient, then glucose is unable to give energy. Glucose is unable to give energy. And in our body, RBCs are such cells which depends on glucose, whether body is in fed state or fasting state or starvation state. They are only dependent on glucose. RBCs cannot use any other fuel for energy. So when glucose is unable to give energy, then there are no ATPs in RBCs. So sodium potassium ATPase pump will be affected, leading to hemolysis. So this is the reason of hemolysis in G6PD and pyruvate kinase deficiency. The reason is different in both the conditions, right? Now let's do the energetics of glycolysis. one glucose molecule gets converted to 2 molecule of pyruvate. Pyruvate is a 3-carbon compound. So from 6-carbon glucose, we get 2 molecules of 3-carbon pyruvate. And here from glucose to pyruvate conversion, there are 10 steps. In these 10 steps, how many ATPs are used? Minus 2 ATPs, that is 2 ATPs are used. 1 ATP used at hexokinase step, other ATP used at phosphofructokinase 1 step. And there are 2 steps of substrate level phosphorylation which are producing 4 ATPs. So I'm writing plus 4 ATPs. So ATP thing I have written on left hand side. On right hand side I will write about NAD, NADH. And you know, in glycolysis there is one step, glyceraldehyde 3-phosphate dehydrogenase, where NADH is produced. So NAD gets converted to NADH, and here this occurs in phase 2. So I have written 2 here. 2 NAD forms 2 NADH. These 2 NADH will later go into ETC to give 5 ATPs. So the full energetics is 5 plus 4, that is 9, minus 2, that is 7 ATPs we get in case of aerobic glycolysis, right? But in case of anaerobic glycolysis, these 2 pyruvate gets converted to 2 lactate, and this is 11th number step of glycolysis, we can say, and the name of enzyme is lactate dehydrogenase. You know, whenever dehydrogenase, then somebody will come to take hydrogen. Mostly NAD forms NADH. But lactate dehydrogenase is an opposite dehydrogenase. Here NADH will form NAD, right? So opposite will occur here. That is, in this step, this NADH which was formed earlier, that will be converted to NAD. So now the energetics of anaerobic glycolysis will be, these 5 ATPs are not obtained because this NADH has formed NAD. So we can't use this NADH for ETC. So these 5 ATPs are not obtained. So energetics will be only this 4 minus 2, that is 2 ATPs. So we get 2 ATPs in case of anaerobic glycolysis, and we get 7 ATPs in case of aerobic glycolysis. Now, one very important question is asked. You can note here that there are 10 steps in aerobic glycolysis, but there are 11 steps in case of anaerobic glycolysis. So they can ask you that what is the purpose of this extra step of anaerobic glycolysis? Is the purpose formation of lactate? No, lactate is just the side product. It is just the dead end of glycolysis. That is not the purpose of this step. In fact, the purpose of this step is formation of this NAD. Why? See, if NAD is not formed, then what will happen? Whenever glucose forms pyruvate, then at the time NAD forms NADH, glucose forms pyruvate, NAD forms NADH. So after some reactions, NAD of the cell will be finished. So now glucose cannot be converted to pyruvate. So we need NAD actually. So this extra step of anaerobic glycolysis is for the regeneration of this NAD so that it can be used again for conversion of glucose to pyruvate. So this step is for regeneration of NAD. Now if I ask you how many ATPs are formed in RBC in aerobic state, what is the answer? Answer is 2 ATP. See how? RBC have no mitochondria. When mitochondria is not there, that NADH which give 5 ATPs via ETC occurs in mitochondria. So this NADH can never give those 5 ATPs in case of RBC. So in RBC, energetics is always 4 minus 2, that is 2 ATPs. In the question, even if they write fed state, fasting state, aerobic state, anaerobic state, they are just using these words to confuse you. But whenever question on RBC, you will say 2 ATPs, right? Now, if they ask you which molecule is dead end of glycolysis, I told you dead end of glycolysis is lactate. What that means? Dead end means once lactate is formed, after that nothing can be formed. Let's see the example. Let's say this is muscle, and let's say this muscle is exercising muscle. This muscle is exercising muscle, and let's say this is liver here. In the center, this is blood, right? Now, during exercise, anaerobic conditions are created so that in this muscle cell, glucose will form pyruvate and then pyruvate will form lactate because of anaerobic glycolysis. Now lactate is the dead end of glycolysis, means when lactate is formed, after that nothing can be formed. That is, in this muscle cell where lactate is formed, in this cell lactate is the dead end, but in body it is not the dead end. That means in body it can be converted to some other thing. That is, see, now lactate has to come out of this muscle cell into the blood. From blood it is taken up by liver. In liver, lactate will form glucose. By which pathway? Lactate forms glucose by gluconeogenesis, and this glucose is again supplied to that exercising muscle. So this is a cycle formed known as Cory's cycle, also known as glucose lactate cycle. Now there is one more similar cycle similar to Cory's cycle, and that is glucose alanine cycle. Let's see what happens there. In case of glucose alanine cycle, let's say this is muscle, and now this muscle is fasting muscle, and let's say this is liver. Again in the center this is blood. So what happens here, in fasting muscle, glucose will form pyruvate, and then pyruvate will form alanine. This alanine comes out in blood. From blood it is taken up by liver. Alanine in liver again gets converted to glucose by pathway gluconeogenesis, and this glucose is again supplied to the fasting muscle. This is known as glucose alanine cycle and also known as Cahill cycle. Cahill, Cahill cycle. So can I say that Cory's cycle occurs in exercising muscle and Cahill cycle occur in fasting muscles, right? Now see one thing here, in liver alanine is forming glucose by gluconeogenesis. This is one amino acid which is forming glucose. Actually, many amino acids, that is 18 amino acids, can form glucose, but you can see here alanine forming glucose. Alanine is most glucogenic amino acid. It is a 3-carbon amino acid, and mostly 3-carbon compounds are good substrates for gluconeogenesis. You can see lactate is also 3-carbon, pyruvate is also 3-carbon, which is forming glucose here by gluconeogenesis. Mostly 3-carbon compounds are good substrates. And alanine amino acid is the most glucogenic amino acid out of 18 amino acids which can form glucose. So now we are going to do RL shunt, that is Rappaport-Lubering shunt, which occurs only in RBCs. Let's see what happens here. First, see normal glycolysis. You know, in normal glycolysis, there is glucose gets converted to pyruvate, and there is one substrate level phosphorylation step occurring here, that is 1,3-bisphosphoglycerate gets converted to 3-phosphoglycerate, right? This step occurs with enzyme phosphoglycerate kinase. It is a substrate level phosphorylation step which will make 2 ATPs at this step, right? And after 3-phosphoglycerate, there are many steps, and then in the end, pyruvate is formed. Now what is RL shunt? In RL shunt, this substrate level phosphorylation step is not occurring. Glycolysis is occurring, just that this step is not occurring. Instead of this step, there is a shunt which is followed. So instead of this straight route, this shunt route is followed. Let's see how. 1,3-bisphosphoglycerate gets converted to 2,3-bisphosphoglycerate, and then 2,3-bisphosphoglycerate forms 3-phosphoglycerate, and after that, after next steps are same and pyruvate is formed. So this shunt is followed, and that is known as RL shunt. Let's see the enzymes here. When 1,3 is converted to 2,3-bisphosphoglycerate, there is intramolecular transfer or shifting occurring. This enzyme is mutase. When 2,3-bisphosphoglycerate gets converted to 3-phosphoglycerate, there is removal of phosphate. You know, the enzyme for removal is phosphatase, right? So RL shunt, that is Rappaport-Lubering shunt or cycle, Rappaport-Lubering shunt or cycle, this occurs only in RBCs, when this occurs only in RBCs, so how many ATPs will be obtained in RL shunt? You know, in RBC, ATPs obtained are 2. Be it any case, ATPs obtained are 2. But in RL shunt, we are losing 2 ATPs because of one step substrate level phosphorylation step of PG kinase not occurring in this case. So 2 minus 2 is 0. So number of ATPs obtained, net gain of ATPs in RL shunt is 0. There's one question in your guide. They ask you that how many ATPs are formed in RL shunt? 0 option is not there, right? So now you can mark 2. Listen to the question again. How many ATPs are formed in RL shunt? So you can say 2 are formed. But when 0 option is there, then best is to mark 0 option. But if 0 option is not there, then you can mark 2, right? See, whenever questions on energetics are asked, then by default you have to consider net gain of ATP they're asking, right? But if this net gain of ATP, this thing answer is not given, then you can consider the other way around, right? So I can say here that full story of RBC is this. The full story of RBC is this, that whatever glucose is entering into RBCs, 90% of glucose out of this is going into normal glycolysis to give you 2 ATPs per glucose molecule, right? But 10% of this glucose, 10% of glucose entering into RBC is not going into normal glycolysis, you can say. It is going into RL shunt, so that this 10% glucose is not forming any ATP, because this 10% glucose purpose is not to make ATP. Instead, its purpose is formation of 2,3-BPG, right? So, can I say that number of ATPs formed in RBC can never be more than 2? But it can be less than 2 if it is RL shunt, right? So what is the use of this compound 2,3-BPG in RBCs? This 2,3-BPG is used for the release of oxygen from hemoglobin A. It can only affect hemoglobin A because hemoglobin A is alpha to beta 2, 2 alpha to beta chains, and this compound 2,3-BPG binds to beta chain and affect the release of oxygen. So therefore, it will not affect any other hemoglobin. Any other hemoglobin like fetal hemoglobin HbF is alpha to gamma 2. There is no beta chain in any other hemoglobin, and 2,3-BPG binds to beta chain. Therefore, 2,3-BPG will not affect any other hemoglobin, right? And also, you know, fetal hemoglobin should not be affected because fetal hemoglobin should take oxygen from mother's blood. It should not release oxygen, right? Now tell me, how many irreversible steps are there in glycolysis? In glycolysis, there are total 10 to 11 steps. Out of them, 3 are irreversible, rest 7 or 8 are reversible steps. So tell me, glycolysis pathway is reversible or irreversible? Yes, glycolysis pathway is irreversible. Even if one step is irreversible, then the pathway is irreversible. So glycolysis is irreversible. Next, tell me, is glycolysis complete breakdown of glucose? No, glycolysis is not complete breakdown of glucose. Complete breakdown of glucose is C6H12O6 plus 6 oxygen will give 6 water and 6 carbon dioxide. These water and carbon dioxide will be released few in link reaction TCA, etc. So when link reaction TCA, etc. also occurs, then it is complete breakdown of glucose when all these 6 carbon dioxide and water are released from glucose, right? So glycolysis is not complete breakdown of glucose. Now let's take the inhibitors of glycolysis. Inhibitors of glycolysis. First is iodoacetate. Iodoacetate will inhibit the enzyme glyceraldehyde 3-phosphate dehydrogenase. Second is arsenate. This also inhibits glyceraldehyde 3-phosphate dehydrogenase. Same enzyme is inhibited. Third is sodium fluoride, which inhibit enolase enzyme. And you know, the sodium fluoride is also used in blood glucose estimation for inhibition of glycolysis, so that when we are measuring glucose in lab, in vitro condition, then the cells should not use glucose by glycolysis, because if cells will keep using glucose in that blood sample, then by the time lab will check the glucose, by that time glucose will be falsely decreased. It will not represent the true glucose value which was there in the patient, right? So therefore we have to use the inhibitor of glycolysis in the sample when we are taking the sample for blood glucose estimation. The fourth inhibitor I am going to write is oxamate. This is not oxalate, this is oxamate, which is inhibitor of enzyme lactate dehydrogenase. So I have told you these four inhibitors of glycolysis. Inhibitor of glycolysis means when these inhibitors are there, then glycolysis pathway will be inhibited, right? Inhibitor means pathway will be inhibited, right? But there is one special thing for arsenate. When arsenate act as inhibitor of glycolysis, then this pathway glycolysis is not inhibited. Glycolysis will continue. The glycolysis pathway will continue. Pyruvate will be formed, but just that no ATP will be formed. That is, zero ATP formed in arsenic poisoning in glycolysis, right? And glycolysis occurs in which compartment of the cell? It occurs in cytoplasm. But after glycolysis, we have link reaction, TCA and ETC. All the other three things, link reaction, TCA, ETC, they all occur in mitochondria. So this is it with glycolysis. We will be looking at link reaction in next video.